I'm Gaya Govind, and I'm here to explain the topic cell lift reactor. Cell lift reactor is also known as an airlift reactor. Cell lift reactor is developed from the viewpoint of controlled flow and intensified circulation caused by the density difference between the riser column filled with air bubbles and a downcomer column filled with liquid only or less air. As you can see in the diagram, gas is parched into only part of the vessel cross-section called the riser. The resulting gas holdup and decreased fluid density causes the liquid in the riser to move upward. As gas bubbles disengage from the liquid at the top of the vessel, heavier bubble-free liquid is left to recirculate through the downcomer. Thus, liquid circulation in airlift reactors results from the density difference between the riser and the downcomer. The reactor is divided into two parts. One, the inner gassed region. Two, the outer ungassed region, which was clearly depicted in the earlier diagram. Mean density gradient between the riser and the downcomer region is responsible for continuous circulation within the reactor. There are many parts of the cell lift reactor, and a few of which are 1. The riser. It is responsible for upward airflow. 2. Downcomer, which consists of degassed media and cells. 3. Base, which is connected to a perforated nozzle blank or a sparger to pump pressurized air. 4. Headspace, which consists of the foam accumulation. And 5. The gas separator, which facilitates the liquid or gas recirculation within the reactor. There are two types of the airlift reactors. 1. External loop airlift reactor. In case of an external loop airlift reactor, the liquid circulation in the riser and the downcomer takes place in two separate compartments. Fewer bubbles are carried into the downcomer, and the density difference between the fluids in riser and downcomer is greater. Therefore, circulation of the liquid in the vessel is faster in an external loop airlift reactor when compared to an internal loop airlift reactor. External loop airlift reactor also provides better mixing when compared to an internal loop airlift reactor. And the second type of the airlift reactor is the internal loop airlift reactor. In this reactor, either a vertical baffle or a draft tube divides the reactor into a riser and a downcomer. Gas holdup and decreased liquid density causes the liquid in the riser to move upwards. Gas disengaged at the top of the vessel, leaving the heavier bubble-free liquid to recirculate through the downcomer. In this diagram, you can clearly see the difference between an internal loop airlift reactor and an external loop airlift reactor. Few of the applications of a cell lift bioreactor are 1. To produce biopharma proteins from fragile cells. 2. To produce single-cell proteins. 3. For mammalian cultures. 4. For filamentous fermentation. 5. For biological water waste treatment. 6. For production of the single-cell proteins, etc. Advantages of using a cell lift reactor are that it is designed with no moving parts or an agitator, which will lead to less maintenance and also will reduce the risk of defects. And it also requires low energy due to the homogeneous distribution of the nutrients and shear force. And it can be easily sterilized due to the absence of the agitator shaft parts. Disadvantages of the reactor are that it requires greater air throughput and higher pressure, and also that it is inefficient to break the foam, and also the absence of the bubble breaker. Here's the poster on the airlift reactor. Thank you.